like to thank the organizers for inviting me here. It's very exciting. I've never been to India, and I have to get used to it, and I enjoy it very much. In my talk, I wanted to combine a number of things. I just wanted to, because there was such an excitement about cryo-electron microscopy, I wanted to show a few slides about this, how we got to this point. And then I wanted to talk about the dynamics of the ribosome itself, which we have investigated with this new technology. And then I wanted to tell you about something that essentially was developed rather recently, about how to get information about continuous evolution of states, or in other words, heterogeneity on a continuous scale. So first of all, single particle reconstruction is something like 42 or 40 years old, and this is a paper that I published in 1975, which is essentially a concept paper. Ultramicroscopy, it's notable, is in its volume number 1, so here I'm talking about the possibility of extending to non-periodic objects the low exposure averaging techniques recently proposed for non-destructive electron microscopy of periodic biological specimens. And the reference specifically is to Unwin and Henderson, and which you might know had come out with a spectacular paper exactly in the same year, where they reconstructed the a density map for the bacteriodopsin, for purple membrane protein. There was a lot of excitement about this because it was the first application of low-dose electron microscopy. The whole idea is to spread the exposure over many, many repeating units. Because of that, the idea excited me to do the same thing with non-periodic specimen individual molecules. So later on, there was this explicit paragraph in a science commentary. If such methods were to be perfected, then, in the words of one scientist, the sky would be the limit. And it's really incredible that now, more than 40 years later, we actually have almost reached the sky, because now we can see atomic structures, near atomic structures of many molecules that we could not see before because they could not be crystallized. So this is our workhorse, which used to be in Albany, and now it's in New York City. Now, of course, it's superseded by these high-end microscopes, these big machines, and Columbia is going to have 3 of them very soon. And so we're going to be in very good shape. So imaging the transmission electron microscope means that we get projection images, and we need to figure out how to get back from there to the 3D structure. There are different geometries that we can use. One of them is related to the CAT scan, where a patient is stationary and the arrangement of beam and a detector is moved around in increments, and then we sort of accumulate images. This is the idea about electron tomography, where we have this stationary arrangement of beam and detector, and the patient, which is a molecule or an extended structure, is being tilted around. And now the single particle reconstruction is based on the idea that we have many, many copies of identical structures. And all we need to do is just take a single snapshot or several of them, but all with a random field of molecules. And then we have to identify the orientations after the fact. And that's the hard part. But in this case, we have an accumulation of radiation dose, which ultimately leads to the destruction of the molecule. Here, we can spread the radiation dose out, just as Unwin and Henderson did, over many, many copies. Okay, so this is now the idea. The first needed to figure out two-dimensional alignment, cross-correlation, 2D averaging. The proof of concept was in 1981 with 40S subunits of HeLa ribosomes. There was already the theme of ribosomes. Ribosomes are just very good to develop this method. You can see very good definition of averages, which came out for the first time. And here's the variance map. No, here's the total average, and here's a variance map. And then one step further, it was very obvious that the whole assumption of single particle averaging was, when you look closer, it was invalid because you had heterogeneity of images. So that was essentially solved at the time by the idea of multivariate statistical analysis of aligned images. When images align, then you can think of them as vectors in a high dimensional space, and then they form clusters, and then here you have a mapping into a very low dimensional space, and then the clusters become visible. So this was a very big obstacle, and I think this was really one of the breakthroughs. And how do we find the orientation? That's now really a difficult part, because if you have an unknown molecule and you have unknown projection direction, you know, where do you start? It's a chicken and egg problem. So one has to solve this with some kind of a bootstrap method. The one that we came up with was the random conical reconstruction, which actually involves two images of a field of molecules, one tilted, the other one untilted. I'm not going to go into the details, but this was now really a breakthrough in figuring out how to get hold of the entire 3D geometry. And here you see the first time this was realized by my postdoc, Michael Rademacher. And another way of doing this is by using common lines. That was a method that was then developed by someone who had been visiting as a student with me, Marian van Heel. So there were sort of two parallel tracks in the bootstrap method. But this scheme, once we had a bootstrap reconstruction, we could refine this, and this is now really universally used, this kind of way. Once you have a bootstrap reconstruction, you can then form from that a library of projections, compare it with the experimental projection, and get refined angles. With these refined angles, you get a refined reconstruction. You know, you go on and on in a circle, and then you get as much as possible in the angular refinement. Okay, so now there are sort of different steps here. You see the increase in resolution. The increase in resolution really has something to do with getting better computers that are faster, being able to process a larger number of images. The instruments became better over time. And so we get sort of steady progress here with 11.5, 6.7, 5.5, and then there was a wall. We could not get beyond this with a non-symmetric molecule. And that wall was posed by the poor performance of the existing recording media. CCD was, of course, already a bad replacement for film to begin with. And the film was really the gold standard at the time. But even with film, we only could get to this place. And then this obstacle was then overcome all of a sudden by the introduction of the of these single electron counting cameras. So we had a situation where the entire mathematical apparatus and computational procedures were developed in place, but they hit the wall. And that wall was removed, and all of a sudden, all the possibilities opened up. Okay, so this is essentially, you can see the comparison between the different performances. The CCD is down here, and this is now the performance of the existing cameras. They have become better even. So what you have to pay attention to is that at Nyquist frequency, we still have a significant portion of signal, and that's really completely unheard of before. Yeah, I can skip over this. So now we get a reconstruction of 2.5 angstroms for the ribosome. This is the ribosome of T. cruzi. It's implicated, the parasite is implicated in causing Chagas disease. So that's where we are right now. Here is a local resolution that goes up to 2.3 angstroms. And we see all these individual features. We see water molecules, and I got very excited about this. I had not foreseen this. So what about capturing multiple states of a molecular machine? How do we capture multiple states coexisting in a sample? Well, very clever programs were developed by Jose Maria Carazo, where Schwarz Shares was as a postdoc, and then later Schwarz moved to the MRC, and now we have this very popular program, RELION, that everybody works with. But there are other programs, maximum likelihood. So maximum likelihood classification, it gets you a small number of discrete states. That's by definition. It's simply the program is laid out in this way. You can say, I estimate I'm going to have 6 classes. It gives you 6 classes. You say, I want to have 10 classes. It gives you 10 classes, and so on. So it really doesn't give you an information directly of what the distribution of these states is and how they are connected. And so I'm going to tell you about something new, which is the manifold embedding, and which goes into the continuum of states. So the ribosome is a processive machine and exists in multiple states. Peptide elongation cycle is a cycle by which one amino acid is integrated into the nascent polypeptide chain, one at a time. So for each amino acid, we go around in this circle. You see there are already a large number of discrete states, and the question is always what is in there between. So here's the elongation cycle explicitly, which involves the decoding steps in which the amino acid tRNA enters in a ternary complex with EFTU and GTP, and then if it's recognized, we get GTP hydrolysis, and tRNA is integrated in the A site. And then this is a very interesting step. This is interesting because it involves a very large motion sort of internal motion of this molecular machine is the mRNA-tRNA translocation. And it involves in sort of a catalytic event that is done by elongation factor G in the GTP state. And after EFG enters this whole the mRNA with the connected tRNAs is moved on by one codon, and then GTP EFG is hydrolyzed, and I mean GTP of EFG is hydrolyzed, and EFG exits. So it's a very interesting event, and I've been studying this for a while with several cryo-EM reconstructions. And the structural distinctions that we can actually recognize by cryo-EM is the tRNA positions. The tRNA is in all its different positions, factor binding states, conformational changes of the ribosome, because there is an intersubunit rotation, there is a closure of the small subunit, small subunit head swivel, and in the position of a very large protuberance, which is the L1 stalk. So all these different things are recognizable, and if you have a large data set and you have them all mixed up, then Reliant is actually able to pull them out of the same sample. And this is really astounding that we have this kind of capability. It's unmatched by any other structural method. X-ray crystallography, maybe sometimes you get two forms of the molecule in the same crystal. But most of the time, you have one single form, which is defined by energy minimization. It might not be a very interesting state. It's just a state that you happen to get out of the crystal. So around 2000, we had this first observation of a very large motion. It's the Ratchet-like motion. It is an intersubunit motion by something like, close to 10 degrees. And it's a very big effect when we bring, when we have the ribosome in the pre-translocational state and add EFG with a non-hydrolyzable analog, then we see this complex here. And it was the first time that this kind of thing was observed. And then you see it already just from the distance that the small subunit has rotated against the other one. This was just very interesting, and it spawned a whole literature on its own later on. So I'll just show you that in a movie. So these are simply two reconstructions. These two reconstructions spliced together. You can see the effect of adding EFG. So the interesting thing here is that there are multiple bridges that connect the two subunits. And what happens with these bridges when you have such a large rotation? Well, they have to, some have to break and reform, and all in, multiple times, like, something like 20 times per second, which is the rate of polypeptide synthesis. So it's a really an incredible event that one has to very closely study and see how it can even be realized. And then later on, we found out that the ribosome is using this not just in translocation, but also in initiation, termination, recycling. Each time you have the intersubunit rotation as part of the functional conformational motion. And subsequently, there was a study by Fred and then single molecule Fred later on, and it turned out that first of all, there was a confirmation that this was actually happening in real time. You could really see that there were distance changes that conformed with the idea that we have an intersubunit rotation. But then a very interesting fact that came out of this study was that we didn't need EFG to get the intersubunit motion. It would also happen, except for a different rate, even in the absence of EFG. And that really is very interesting. Well, first of all, what does this rotation mean in a functional sense? Well, it is really, it makes a lot of sense in terms of getting the mRNA moved by one codon. You can just think of mRNA being locked to the small subunit, and now you rotate it. You rotate the small subunit, and then if you had the mechanism to unlock the mRNA from the small subunit and have a reverse motion of the small subunit, then you get exactly where you started, except you have advanced one codon. Okay, so it really made sense in a beautiful way. So, but from the other observation, we figured out that this is really something that the ribosome is doing all by itself in the thermal environment. And a ribosome is built in such a way that it picks up the energy in a functionally meaningful way. And the intersubunit rotation is facilitated by the architecture. There are tight RNA bridges in the center and loose bridges at the periphery. So then, so I called it Lucretian motion instead of Brownian motion because Lucretius has actually talked about this. Observe what happens when the sunbeams are admitted into the building and shed light on its shadowy places. You will see a multitude of shiny particles mingling in a multitude of waves. The dancing is an actual indication of underlying movements of matter hidden from our sight. It originates with the atoms. He used the term atoms, which move by themselves. Okay, so he had it all figured out, and Brown shouldn't be credited with it. So if we use a model, a very coarse-grained model of the ribosome, and subjected to normal mode analysis, it actually performs exactly this motion. This is one of the principal motions. So I compared with locomotion, human locomotion. The legs, act as pendulums, and we move, and we don't have to exert a force to get one leg in front of the other. It sort of swings. It swings along. Okay, so it's a very similar leg. So the translation, the ribosome is built to rock, and the tRNA goes along like this in these kinds of steps. All right. And here are the tRNAs that are inferred from cryo-EM maps. You can see the individual steps there. So as these kinds of programs became mainstay, many people now saw multiple structures from the same specimen. Here we have the mammalian ADS pre-complex. So all the different motions that are implicated in function, this head swivel, small subunit head swivel, intersubunit rotation, the L1 motion, they're all seen here. And then along with it come the different positions of the tRNAs. This is where we had the first use of a of such a maximal likelihood program, and this was to a pre-translocational ribosome without EFG. So it was very clear from this here that all these multiple states coexisted there. The molecules in there, they were simply freely exerting their freedom in the thermal environment. And so it's no surprise that if we now take a cell, well, what I showed before, these were all in vitro states. So it was a defined translation system. But then you figure that if you take an in vivo system, you take cell extracts and purified ribosomes from cell extracts, you would find them randomly in all different states. And that's exactly what came out of such studies. So this is a cell from a cell extract, and we have all these multiple states, and for some reason, which has to do with the purification and also the occupation of the states, the duration of these states, we have them represented in different quantities, and some of them are completely absent. But you can understand the excitement that is now in the entire field, that we can take a sample from a running molecular machine. You can extract them. You see all the different states. Now you can connect them. And I called this story and a sample. You have a story to tell. You see all its pictures. Now you have to somehow connect them with arrows. Yeah, this is another study like this, where we see a discrete number of these states. Here's another one with the eukaryotic ribosome. So now a little bit of theoretical considerations that come from it. The energy landscape is expected to be fine-grained. You have this very large motion, and on the other hand, you need to go from here to there without losing contact. So a fine-grained texture is required for processivity of the ribosome and high fidelity in translation. Any missteps are not allowed. So there is a very interesting paper by Monroe et al. And Tips, Scott Blanchard's group, Conditions for the Function of a Processive Molecular Machine. The energy landscape navigated during translation is likely to consist of a multitude of conformational substates, each with relatively similar stabilities. And here, so he's talking now about the barrier heights. It has to be in such a way that under a thermal environment, we are able to go from one state to the other. We cannot have any very low energy in between, because then the entire system would be trapped, and the life would come to a standstill. Yeah. So they distinguish in this theoretical paper between macroscopic states, mesoscopic, and microscopic states. But implied in it is that there is really a continuum at each level of graininess, and that you never have an energy minimum that would stall the system forever. They specifically about the ribosome, they say structural snapshots of the ribosome represent only the most thermodynamically stable native state conformation of the particle. They likely represent only a small portion of the many potential conformations of the ribosome transited during the process of translation. That simply means the reason why we have not observed these states is a sampling problem. If the data collection is large enough, and the number of classes we specify is large enough, we could say 100, 200, in maximum likelihood calculation, then we would see them all. But nobody ever specified so many classes because the computations could, of course, exponentially up. Okay, so this is sort of an something that I try to approach in a collaboration, the long-standing collaboration with Abbas Urmaz. It's a theoretical physicist in Wisconsin. So how do we get information about the continuum of states? So we have to assume that multiple conformation and binding states coexist. There are several degrees of freedom. The states often form a multidimensional continuum. And some states are preferred. It simply means that they're most highly occupied, most frequently visited, but all the others are there as well. So we did have the rely on, but it doesn't answer the question how are the most highly occupied states connected with each other. The idea is if we collect enough data, then we will find them as well. And then we hope to find trajectories of states. Okay, well, I already told you there's a strong evidence for continuum. So let's say I just want to represent one image by one point here, and I have this two-dimensional continuum. Let's say rely on looks at it, and it would say, well, there are more in this environment than anywhere around, and maybe there too, but it wouldn't pick up anything in between, but these states all exist, okay? So mapping of continuous changes, there's Abbas Urmaz, Ali Dashti, Peter Schwander. These are the people in Wisconsin, and these people on my side, Cao Liao and Suprajit Maji. A manifold is simply an entity in an n-dimensional space. Aligned images, if they are similar, they form a cluster in n-dimensional space, because similarity translates into proximity in that space. And continuous changes in conformation of a molecule produce a certain arrangement of vectors in this continuum, and it's naturally in some kind of a subspace. And this really goes back to something that I developed a long time ago, together with the student Van Heel in 1981, based on exactly the same principle, the clustering of aligned images if they represent the same object. So topological manifolds, these are sort of definitions, and I probably don't have to tell you about this. Here's the Earth's surface, and the local patch is planar. So in order to describe this geometry, we don't have to go into the three-dimensional space. We can stay in the two-dimensional space, and that's all that's interesting in terms of describing the local environment. And obviously, this led to the belief that the Earth is flat. If you look outside and you see just a two-dimensional arena, then you wouldn't think of the curvature. But the point is, it doesn't matter that this is now in 3 dimensions. It's totally sufficient to describe it in 2 dimensions. And this here indicates exactly the same thing. If you have conformational heterogeneous molecules, then you have a subset of images in each projection direction that forms a point cloud that represents the manifold. Where any projections of a molecule, you take a particular Eulerian angle, then you see all the conformation from such a manifold. And the characterization of this manifold requires a mapping that follows the topography of the manifold. Okay, so here it is indicated by some kind of a sheet, a sheet that is in this higher dimensional space, and to describe the relationships of the images on here, it's completely irrelevant to the way they're arranged here in this higher dimensional space. What we need to know to do is to describe exactly this here in following the local geometries. Yeah, so we have to unroll this, and this is done by this manifold learning process. We have to embed this in a low-dimensional subspace. It's indicated by these guys putting in different directions. So we have to aim to preserve the geodesic distance and not the Euclidean distance. Here's an example for the use of such a procedure. We have images of faces in different postures and in different lighting conditions, and with manifold. Thank you. So with a manifold mapping, you can get this kind of spread of faces, and you can see that despite the very large variation in so many features, this kind of approach has led to a completely realistic ordering. Okay, so I just jump into the experimental example that we used. We used 850,000 particles of yeast ribosomes purified from a cell extract. When I say purified, there was really no precaution for, leave the tRNAs in place and be very careful and so on. These are simply purified. And so we don't expect them to be in a particular functional state. So the whole approach treats classification as a two-step process. We first sort by orientation in a conventional way, and then we map the conformational changes within each projection direction by the manifold embedding approach. In fact, this is an old image where we just simply produce averages for different projection directions, and at the same time performed the computation of the associated variance map, which means it has very high spots where there's a lot going on, where there's a lot of differences between all the views that fall into direction. You can see there's a lot going on. It simply means, for instance, for all the images that have been thrown into this particular orientation, they differ. They differ by a lot. Oh, Jesus. Okay. Okay. All right. So the recipe for mapping of states, we classify single particle data first by orientation, And for each projection direction, the data reflects the existing multidimensional conformational spectrum. Now we perform manifold embedding or diffusion mapping for each projection direction and do an eigenvalue decomposition of the data in the new space. And it gives us a low-dimensional space of reaction coordinates. And now this is now a very important step. For each projection direction, we have this mapping individually, okay? And but now, how are they all connected? How can we put all this information together? And my collaborators found a way of consolidating this into one single map. So I cannot tell you this in any detail. I refer you to the paper that we published in 2014. But here, excuse me, first that one here, okay. This is one single projection direction. If we just look at the ribosome data, the projections in that direction, you have to think about, this one has to be added to that one. This is the average of all of them. You have to add this to that one in order to get the individual configurations. Okay, now we do the other one. Okay. Now, if you look at this one here, this is really the highest ranking eigenvalue. It gives us the most important reaction coordinate. And you can see this boundary here. What is happening right here? Well, this is the boundary of the small subunit. And this here represents the intersubunit rotation. You can immediately pick this up. So this intersubunit rotation in this very large data set was the most prominent one. So this is just an example from one particular projection direction. So altogether, this is the flow diagram from the start to finish, where we get, in the end, where it says patch information from different orientations. Then we get a mapping of all the occupancies of the different, in a continuum of states. And from there, we can use a Boltzmann formula to compute the free energy landscape. So this is the first experimental method that gives us energy landscapes of a molecule. So high occupancy translates into low free energy, low occupancy and high free energy, which means states that are difficult to reach at biologically relevant temperatures. So now we can go into this landscape and just grab an environment, a local environment, and make a reconstruction from it. So we have local reconstructions of local states in a continuum of states. Okay, so now this is the energy landscape that we get from this large data set. In blue, this you can see that we see a closed trajectory, which indicates that a molecule that is right here somewhere, has this way to move. So this is the trajectory available to a molecule. For each, you can see, I hope you can see this little dot moving around. For each position, we have a local reconstruction. You can see all kinds of changes, one after the other. I have another one like this. I think this is another movie. And in this case, we're just moving around at these spots here, and not in the continuum. And each time we show in the different views how the ribosome changes its conformation. Now, the very interesting thing is it's already hinted at by the closure of this whole path. It's already that the conformational changes that we see are reminiscent of the ones that had been previously obtained by cryo-EM for discrete states. And they really represent the changes of ribosome structure at the different stages of the elongation cycle. So, which is intersubunit motion and the closure motion of the small subunit, the head swivel, the L1 movement. Everything is visible here. And moreover, they are actually arranged. They're arranged in the manner of the elongation cycle. So this result is in a sense really baffling, because we cannot claim that the ribosome is doing anything useful. What it tells us is that all these molecules that are in the free thermal environment are actually doing motions that they would do in a functional context, and which seems to be just an extension of the idea of the intersubunit motion. It's only extended to more degrees of freedom. So all these different degrees of freedom are actually being played with. When I first saw this, I thought the ribosome is dreaming. Okay, it's dreaming of doing some useful work, and it's going through all these different motions. So you have a free energy trough along a closed path. Domain motions kept that are recognizable from previous studies, and I just said it before. This is the most recent use of that technique, and it just shows you just one single projection direction. It's a very large motion that is produced by ribosomes which have an unusual mRNA. They have a non-capped mRNA, and they have some kind of a pseudoknot that produces obviously conformational contortions. And just to show you how useful this technique is, I show it to you here in the context of the calcium release channel. This has nothing to do with the ribosome studies, but this is the most recent application of it. This shows the calcium release channel, which is supposed to open under the influence of a very small dose of calcium and produce a big gush of calcium and causes muscles to contract. And we want to see how this opens and closes. Well, there are now two energy landscapes. One is without ligands, and the other one is with ligands. And we can compute the transition probability that leads from here to here. That transition probability has a hotspot right here, and right here. So it tells us that there is no chance for the molecule to get from here directly down here. It has to go through a low probability pathway until it reaches the hotspot and then goes down here. And here it lands at a place which is again not the open state, but it has to find its way into it. Okay, so you can see now we can analyze a molecule, the workings of a molecular machine, by experimentally obtained energy landscapes. And so I think this is the true future of cryo-EM. Right now, the way the data are analyzed is really akin to, we're thinking of them as static molecules, and that's the way X-ray crystallography approached molecules. And this is simply because crystals can only contain one particular form of a structure. So in summary, and this is now the summary of this new method, because I gave you the summary of the other one before. This is a true ensemble method to map multidimensional continuum of states. The path to ligand binding is through a range of conformations visited by the unbound molecule. Well, that's, I forgot to say. In the upper landscape, there were no ligands present. And the only way how this molecule reached a state in which it could even accept ligands was through going through multiple conformations, and including low probability conformations. So the method is generally suited to map trajectories of molecular machines, and it lends itself to exploration of the effect of parameters like temperature, buffer conditions on the dynamics of the molecule. You can change buffer conditions, make the same mapping again, and look exactly how it influences the energy landscape, and overcomes the deficiencies of maximum likelihood method. And it overcomes the idea of molecules as a set of static structures. And then I have to emphasize, this gives us a continuum of structures that has nothing to do with the morphing between two structures. Morphing leads us to a completely unexplored pathway. It's completely postulated, and it has no experimental support. Okay, so this is my group presently on top of the building. It's called the William Black Building, 168th Street. We're looking down towards the south. And every summer we assemble up there, and be it hot or raining and so forth, and take a picture. So that was this year's picture. Okay, thank you.
Robert, when you are generating these states to superimpose to get this landscape,
yeah.
So how many states or how many configurations you have need to solve structures?
Well, I mean, they are really, the continuum is very large, but obviously only the ones on the trajectory are actually meaningful. They are observable, and there were 50 there. 50.
So, but these are like the structure was solved at a low temperature, cryo-EM. So what is causing this conformational change at that temperature?
What causes it?
Yeah.
Well, these are molecules that are free to assume all different conformations.
Even at that low temperature? Even at that low temperature?
Well, it's not low temperature. It's room temperature.
It's room temperature.
And then you freeze it quickly, and the idea is that it maintains exactly the structure that you saw at that point. And hopefully nothing happens in between. And nobody has investigated this. So we can be off by something, but you do see that lots of things are going on.
Just one here, Joachim.
Yeah.
Thanks for beautiful talk, actually. You talked about ratcheting relating to the translocation of a codon. There are substates of ratcheting also. So does it mean that actually the translocation is happening nucleotide by nucleotide?
That
the translocation is actually happening nucleotide by nucleotide, one nucleotide at a time.
You mean one?
So the ratcheting.
One base at a time, or?
Yeah, one base at a time.
No, not necessarily. It really needs to span the length of one codon.
Correct.
That really is defined by certain registration points of the tRNA, and these in between states they have to solve the problem of guiding the two molecules next to each other and keeping them in registration all the time, so that there's no slippage in any place. So it involves many different, salt bridges and so forth, many different substates. Well, we had initially looked at this with maximum likelihood method, and we did see one additional substate. So I can't recall how that was defined now structurally. We faced the interesting question together with Debashish and Ruben Gonzalez. Ruben Gonzalez performed single molecule FRET experiments on exactly such a system, and he didn't find this intermediate state. And then we posed the question: under what conditions can we see something in one experimental method and not in the other? And the answer, I think, is simply because the time window is too wide in the single molecule FRET. So you could have a state which is highly populated, but it's only visited for a very short time, and then they would be totally invisible for single molecule FRET. Yeah.
So if I understood this method correctly, you do this manifold embedding for every projection direction separately, but then to get this energy landscape, you have to somehow bring those together. And
yes.
So the problem I see is that you have to somehow identify which structures in this continuum in different projection directions are the same.
Yes, and the answer is really, I think this is an ingenious idea. If you think about the occupancy of states, the occupancy of states should not depend on projection direction. Mm-hmm. So if you have a histogram or some kind of a generalized histogram of the distribution of states, you can match the histograms, and so that's how you can get the entire all the data merged.
I see. So basically, you use the consistency between the different directions as a criterion to.
Well, it's a consistency. You postulate that the distribution of conformational states does not depend on the view angle. Mm-hmm.
The question is, how important is the choice of the collective variable or the reaction coordinate that you have decided to choose? So how does the manifold that you're talking about, the non-linear manifold, emerges with the choice of the reaction coordinate or the collective variable?
These are simply the highest ranking ones. Typically we have a five-dimensional space, and then we somehow go into noise. The five-dimensional space is typically here for 5 degrees of freedom, and we are only depicting the first two, the most dominant ones.
Additional, so you showed that Swiss roll figure.
Yeah.
And so if you have gaps in the Swiss roll, this process does not work, right? It does not open up like you would.
Yeah, it's
if there are holes in
it's very difficult. Yeah.
And that would mean that you need more and more samples to actually.
Yes.
Thanks.
So one question that I had, more of a naive question. You said in the beginning that the current resolution limit that we have is 2.5 angstrom.
That's just what we obtained with the ribosome, and I think that's still unmatched by any other group.
Oh, okay. Is there a need to go below that? will we learn?
Well, the best cryo-EM resolution that has been obtained is really at 1.9 or something like this by suprammanium for a different molecule. So it's really, but it gets more and more difficult because there's so many errors that can come in between, and you know how Fourier space is. It's unforgiving, you know. You get more and more. You need more and more space out there, so you need a lot of information.
But presumably, is there additional information hidden there, which is?
Well, I think the most important hurdle is really the heterogeneity, because if you have heterogeneity, then you might have to do data collections that are simply beyond feasibility.
Sort of decipher the unstructured regions of the protein using this. Can the unstructured region of the protein be sort of visualized using this continuum of space strategy, or is it only applicable to a very structured part of the protein which is not?
Well, there's no principal reasons why they cannot be used, but there are practical reasons, because the unstructured ones are most likely to flop around, and so they are in multiple conformations. So in order to observe any subset, you have to go into millions of data collection.
Yeah, that's what I probably saw in one of your pictures, the tRNA.
Ah, you looked at it, right?
Yeah.
Any other question? Okay, if not,
Thank you.